Um, so I currently head BIOS, and prior to joining Bio, I was the head of robotics at one of India's top robotics labs, and we developed Lampy and a few wearables over there. I did my PhD in Hong Kong, and in Hong Kong, I had the opportunity to set up a foundation model team, and this team consisted of folks from Alibaba Quen and Unimax. So what we focused on was building a foundation model that runs on this robot that you see on the screen. So we called it Lampy, and it's essentially a robot that cannot speak, but it has to emote like an animal. So the reason I bring this up is because I'm bringing transferring those learnings into BIOS. Um, in my PhD was in biomedical engineering, so I'm bringing all this expertise of robotics and foundation models into BIOS, focusing on building the wet labs of the future. So through this presentation, you will see what we are doing right now at Bio and what we have in store for the months to come. So, one of the initial statements that I would like to make is both coding and mathematics, the verification is very fast. So, that's the reason you see models like Fable be extremely good at coding and math. But in biology, the verification is in the real world. You need to run tests, you have to run an assay, and you have to wait for weeks, sometimes much longer. So our goal with the wet lab of a future is, if we bring robots into the lab, how fast can we theoretically go? So here, what you see in front of your screen is an example of cell culture, one of the most common things done in a wet lab. And it usually takes 3 to 4 weeks, all these processes. And one of the most time consuming aspects, as you can see, is watching these cells grow and the recover phase. It can take as long as 24 to 48 hours. Automating this could cut down the time by half. So this remains unchanged because that we need to observe what the cells are, how they are changing, but the rest we can accelerate time and save a lot of time. So that is what we are aiming essentially to do. So at Bio, currently we are focusing on drug discovery, specifically binder design. And we have something live right now on BIOS that lets you take a target of interest, a receptor of interest, and we have an entire workflow end to end within BIOS that lets you discover binders for this receptor. So I'll just quickly show you how that would look like. So you go into BIOS, you query, let's say GLP-1R. You can see the entire sequence from AlphaFold DB and UniProt, and we have a whole stack of techniques to annotate it. And once you have the binding sites, we have a set of models that start generating the candidates. So if I go, so here is one candidate which is sort of like a binder design. So we have models running on the GPU that are running live right now, generating a binding candidate for GLP-1R. It's essentially, you can think of it as it's creating a drug uh, in real time on the GPU. Obviously, you have to validate this, so that's why the wet lab and robotics angle comes into play. So right now we have all the state-of-the-art models on BIOS, which are BoltzGen, RFDiffusion 3, ProteinX design from ByteDance, BindCraft, and Genie 3. So just to give you some perspective, the last model that you see, Genie 3, was just launched like 2 to 3 weeks ago. So at BIOS, we make sure that the state of the art and latest models are always available and readily accessible through BIOS. So that is one of our unique aspects that we offer within BIOS, that we are super proud of, that we ship extremely fast and we have the latest and greatest on BIOS. So the whole aspect that I spoke to you, so my colleague Johannes will be going in depth in the next presentation about this, but it's in silico design. So we computationally use GPUs to generate binders for a target of interest. So it could be a well-known target like GLP-1R, or it could be not so well-studied receptors like HIS1R receptor. We have a whole scoring mechanism which is chemistry based, physics based, and structural integrity based. What I mean by that is we do molecular dynamic simulations to identify if the generated binder actually docks to our receptor of our target receptor. So it's an extremely, we scrutinize these generated candidates, right? We get 1,000 candidates in a span of an hour or two. And I think something that should be very seriously considered is we can generate 1,000 candidates for $30. So imagine generating the future medicine for like $30. That's it's sort of not possible any time in history. So we are at a very unique point in time of history where we're going to see personalized medicine happen, and robots come in and truly accelerate this. So at BIOS, we want to be the place where people come. It can be both seasoned biologists, but also just biohackers who are interested in experimenting. They can come on BIOS. They can give their blood test report and generate personalized binders. So that is what we want to enable. Like I mentioned, if you are generating about 120 verified and vetted designs, it can cost like $10, and it can be done in an hour. So coming to the next, some exciting features within BIOS is we have computer vision in BIOS. So what this means is, let's say you're a bio lab, a wet lab that's trying to study mice. Uh, if you give alcohol to mice, and if you don't give alcohol to mice, how do they react? So if you look in the middle, these mice are given, they're old mice with alcohol and they barely move. They just want to sleep. But young mice in alcohol move a little, and they developed a new drug called as new limit, and with that, even if you consume alcohol, you can still be mobile. So but to track this behavior, right, you need systems that can annotate it, segment it, and in real time process it in a GPU. So that is what we enable in BIOS. A biologist can come, put in their data, put in their video, they can select multiple targets and essentially track it. Another example is, here there is a immune cell chasing a bacteria. So today it is possible to live track any kind of bio footage within BIOS. What this essentially, so the first part that I showed you, the in silico design, and now the computer vision, this all culminates into the hardware aspect. So at the end of the day, a predicted binder is still just a hypothesis. So that's why we are currently putting efforts to build hardware. So bringing my expertise from having built robots in the past, several robots, we are bringing it into bio, and super excited about it. So I'm happy to introduce Robios. Robios is a one of its kind autonomous lab in a box. So it's one arm. It can do many experiments. Essentially, the goal with Robios is it can synthesize these peptides. So we generate binder candidates, and then Robios essentially allows us to generate synthesize these peptides. So the first thing is a robot in a box, and the next one is something that can validate this. So if we have to run assays, and we have to watch if the binder is actually binding to a receptor on a cellular level, and if we have to do cell culture, we have to test these peptides and how they are functioning. We essentially need these two kinds of robots. So that is what we are working on. What you see right now is a simulation. So what I want to get into your head is that in robotics, You guys might be aware of the concept of reinforcement learning. So what this means is, this robotic hand, we train it in a simulation on how to deal with pipettes, beakers, well plates, and then we essentially, it learns how to interface with these medical tools in the simulation, and then when we bring it to the real world, it sort of transfers the learning from simulation to the real world, because the robot still thinks it's in the simulation. It's not aware that it's in the real world. So it goes through millions of iterations through reinforcement learning in the simulation, and then it brings all those learnings into the real world. So that we are at a unique position where at BIOS we are doing foundation models, we are doing robotics, we're doing reinforcement learning and computational biology. We are essentially highly deep tech at this point of time, and we want to be one of the key players when it comes to enabling seasoned biologists and normal folks to make real discoveries, real scientific discoveries, all within BIOS. And essentially, what this enables is, we design in silico on the computer, we synthesize it using Robios, like you've seen, our autonomous lab in a box. We test it in assay, as you've seen with the robotic arm. And finally, we take the learnings. We look at which binder candidates passed and which failed. And the ones that passed with, let's say, 90% affinity, we take it back into the design. We feed back it to the system so that in the next run, we regenerate new binders that are more like the most successful candidate. So we get even better candidates. So essentially, there's a feedback loop, the first of its kind, where in each cycle we get a better and stronger binder, and we get a better and stronger candidate. And yeah, our whole point is, we believe that in the future, just like how everyone has a 3D printer in their home, they're gonna have a peptide synthesis machine in their home. So that is the future we look at, and that are the machines that we will build today. Thank you, and I hope you guys like the presentation. Yep.